the special sense of vision is detected in the eye. So let's take a look at some of the structures associated with the eye that helps it to function to allow you to see. So first of all, the eye is protected by eyelids and eyelashes. These structures prevent debris like dust from entering the eye. The palpebral conjunctiva is a thin membrane that lines the inside of the eyelid and covers the cornea of the eye. If you've ever had pink eye is also called conjunctivitis, and it has to do with an infection and inflammation of the conjunctiva here. Your eyes constantly produce tears, and they're produced by a gland that is superior to the eye called the lacrimal gland. The tears then wash across your eye in order to help wash away any debris that might have landed on the surface. From there, the tears drain through the nasolacrimal canal into the back of the nasal cavity. Once it's in the back of the nasal cavity, typically you either blow your nose to get rid of it, or you just swallow it and your stomach acid takes care of any debris or bacteria that may have been transported in the process. The extraocular, also known as the extrinsic muscles, are the external skeletal muscles that control the movement of the eyeball. So they allow the eye to be moved up and down and left and right, and when you roll your eyes. There are three layers or tunics of the eye. There is the fibrous tunic, which is the outermost layer, and so it's the most superficial. And it is a tough fibrous tissue, and it's made up of the sclera, which is the white part of the eye, and the cornea, which is a clear extension of the sclera that is right at the front of the eye that covers the lens and the pupil and the iris. The next deepest tunic is the vascular tunic, which is also known as the choroid. And the choroid is where the blood vessels that deliver nutrients to the eye are found. And then inside or deep to the choroid, you have the retina. And the retina is the sensory layer of the eye. This is where photoreceptors for vision are found. Now inside the eye, you also have smooth muscle, and you have two structures that are kind of attached to each other. One is the ciliary body, and it is posterior to the iris. The ciliary body is attached to the lens, and it holds the lens in place. And when the lens needs to bend in order to bend light so that it focuses on the back of the retina, it's the ciliary body that actually tugs on the lens to change its shape. The iris is the colored part of the eye. So if you have blue eyes, you have a blue iris. If you have brown eyes, you have a brown iris. If you have green eyes, you have a green iris. The iris is also made of smooth muscle, and it can either constrict or relax in order to change the shape of the pupil. So the pupil is just the hole in the middle of the eye. And when you're looking at someone's eye, the pupil is just the black dot in the middle. But it's not a solid structure. It's actually a hole that light is passing through in order to get to the retina at the back of the eye. And so the iris can either dilate the pupil and make it bigger so that more light energy can get into the eye, or it can constrict it so that less light energy gets into the middle of the eye or into the eye to the retina. There's also fluid in the eye. The anterior compartment of the eye is filled with a watery fluid called the aqueous humor. The fluid in the back part of the eye is a little bit more gel-like, and it's called the vitreous humor, and it helps to hold the retina up against the choroid. This ensures that the sensory cells in the retina are getting nutrients from the blood vessels in the choroid. Now, if you've ever heard of someone having a detached retina, There are certain things that can cause this retina to become separated from the choroid, and that typically can result in blindness if it's not fixed very quickly. But they do have a surgery that they can do to reattach the retina. Now, if we look at this picture here, this is what a doctor sees when you go to the eye doctor and they shine a bright light in your eye and they're looking into your eye. They're looking at your retina in the back of the eye here, and you can see these blood vessels in the choroid. And then this area is also the retina. Now, this area off to the side is called the optic disc, and you'll notice that the blood vessels are actually radiating away from the optic disc. The optic disc is a hole in the retina that serves as a passageway for blood vessels and neurons to enter and leave the eye. So there aren't any photoreceptors here because it's a hole, and because of that, this is called the blind spot. And normally you don't notice the blind spot, but sometimes you can force yourself to basically not see something, which we will actually do in lab. The photoreceptors in the retina are stimulated by light energy. So basically, light energy coming from the external environment triggers some sort of change in the photoreceptors. And from there, that information gets sent through sensory neurons through the optic nerve to the brain, where the brain perceives the whatever the object is. There are two different types of photoreceptors. The first one we're going to talk about are rods here. And rods are more sensitive than cones, which are the ones here. So they're more sensitive, meaning that they're more active in low light. So you can sync your night vision to your rods. So when light is low, you're using your rods primarily to see. And one thing with rods, though, is they cannot detect color. So you've probably noticed that at night, when you don't have like an artificial light source with you, you can still kind of see the shadows and the outlines of objects, but you can't tell what color they are. They're also located in the periphery of the retina. So the rods are actually only detecting light energy that's hitting the periphery of your eye. So they contribute to your peripheral vision, but they don't really work if you're looking right at something. So the next time you're in a dark area, as your rods begin to kind of become more sensitive and start picking up light, you can test this out by trying to look straight at something, and you'll notice that you can't see it. But if you try to kind of look at something with your peripheral vision, you can actually see it better. Cones need more light energy in order to be stimulated, so they have to, or they only are active in brighter light. And they do detect color. And there are actually 3 different types of cones, and they detect different wavelengths of light. So you have one cone that is specialized to detect light that is in the red wavelengths zone. You have another type of cone that detects blue, and another type of cone that detects green wavelengths of light. The other colors that you see, like yellow and purple and orange, are really, they're really made in the brain because we don't have a cone for yellow. But whenever yellow light enters the eye, it triggers a specific ratio of green cones to red cones, and our brain perceives that as yellow. Cones are also required for visual acuity or clarity. So for example, if you're trying to read something, you have to look right at it, because if you're not looking straight at it, the image from the text is not reaching the cones of your eyes, and you won't be able to read it. So the cones are directly posterior to the pupil. So you have to look right at something so that the image hits the retina where the cones are so that you can actually make out some of the finer details. We're not going to watch this video on this video, but I would recommend that you open it up in the PowerPoint itself. And you really only need to watch the first minute or two of it. The rest of it actually covers some different stuff. But it helps you understand how we see and how we see those intermediate colors. So how do we see the color yellow if we don't have cones for yellow? So really great video. Watch the first couple of minutes of it and you will probably enjoy it and learn something in the process. Now, coming back to our picture of the retina here, remember that we mentioned in the periphery, this is where we find the majority of our rods. This area right here is directly posterior to the pupil, which is that opening in the iris. The area overall here is called the macula lutea, and the very middle of it is called the fovea centralis. The fovea centralis has the highest density of cones in the eye. So in order for you to be able to read something or make out fine detail, the light energy from the object has to hit this area so that the cones are activated. The macula lutea also has a lot of cones, but it's starting to have some rods too. So basically the fovea centralis is only cones. Macula lutea is mostly cones with some rods, and then the periphery out here is only rods. So basically as you move away from this point, the cone density decreases, but the rod density increases.